Okay, good morning. This is Josh Wagner with Montana Department of Agriculture and the Noxious Weed Early Detection Rapid Response Program. And we'll go ahead and try to kick this off on time because I know Tim and Working Dogs are going to have tons of great information to share. And I am just going to give a quick introduction to kind of outline what's happening, and then we'll hand it to them. So my program is going to be doing EDRR webinars spring and fall. And this year we've got the spring 2023 international webinar. And I think you're going to see a lot of how we collaborate to make this program a success and to make weed management a success. I'm not an expert at very many things, but I know people who are, so you'll see how we use that today. We're going to be discussing Palmer amaranth and working dogs for conservation. So the first introduction, working dogs for conservation. The picture on the left is Alice Whitelaw. She's the co-founder of Working Dogs for Conservation 25 years ago. And she is the point of contact for developing new programs, discussing what weeds could be worked with, things like that nature. In the middle is Fenton, and that's him in Sweetgrass County looking for Dyer's Woad. And I have a special spot in my heart for cow dogs, so I really like him, even though I've never actually met him. And on the right is Ngaio Richards. She's a forensic and field specialist. She's Canadian, which is part of why this is an international webinar. She's a world traveler, very accomplished, and she and I have become fast friends, and we pretty much talk about dogs and weeds and work almost every day, I would say. Dyer's Woad is kind of the big Montana weed success story, and I won't go into it very deeply, but Working Dogs has been a big part of that. You can see we know exactly how many plants we have in Dyer's Woad plants we have in Montana, 418 plants in 2022. And in the 1980s, we had 62,000 plants in one site in one county. So that was plants pulled, 6,000 plants a year on average. And now, thanks to Working Dogs, among many other people, but we're down to 418 plants. And to go back to the previous slide, 10 plants in Flathead County, 12 in Missoula, 15 in Beaverhead, and 380 in Sweetgrass. So we have had some real success. This is my list of 16 weeds that we're kind of focusing on. It's a fluid list and can change, but at the moment, this is it. And Working Dogs is going to go through this list today and kind of talk how their program could help with some of these. And then you'll see the last two weeds, Palmer amaranth and waterhemp. I chose that for a topic this spring because in January, we had a Palmer amaranth and waterhemp task force meeting in Sidney, Montana, which Marley Manukian in Richland County Extension did a huge part in putting on, and we had a lot of people come, and it just seemed like something that we have some momentum going on. It's an important weed, and we should follow up and present to a wider audience. I have a North Dakota State badge there because they're already dealing with it, and we're learning a lot from them. They're one of the experts that we're counting on. So then Tim Seipel is going to talk about that from Montana State University. Tim's probably one of the best botanists I know, and he's our Montana cropland expert, but he's just an excellent ecologist all around, as you can see from these pictures. He's not limited to that. He's a fantastic resource for Montana. And then the last slide I'm going to do before passing it to Tim is It's also the international webinar this year because Melanie Kelly from Tasmania reached out and was interested in the webinar. And they are working on orange hawkweed in New South Wales, which, as everyone knows, we're dealing with here in Montana too. And that's her and her dog, Fonzie, which is a very American name. But I just thought it was really cool. And that landscape kind of looks like Montana. And I actually went in a huge vortex last night of reading about orange hawkweed in Oceania. And with that, I will pass it on to Tim.
Thanks, Josh. Would you mind stop? Yeah, there we go. Thanks, everyone. While I'm sharing my screen, I will say I worked with Keith McDougall in New South Wales. He's retired now, but he on some of the initial early detection rapid responsive as part of a research group that I worked on during my PhD. So it's good to see that it all comes back around. Okay, I'm going to share my screen, and unless I hear otherwise from anyone, I'm going to assume you could all hear me and all see my screen well. I have about 30 minutes, and I probably have way too many slides, so I'll probably breeze through some of them pretty quick. We're going to talk about water hemp and Palmer amaranth today, their biology, prevention, and the potential for spread in Montana. So the taxonomy of pigweeds, we're going to review that in a little bit about why these two species are special. We'll talk about IDing, their biology. Their big risk in Montana is because of their herbicide resistance, what the current distributions are, and what actions we should take for prevention and management of each one of these. So we're really in the early detection rapid response phase for these two weeds in Montana. There are two species in the genus Amaranthus. They are water hemp and Palmer amaranth. Be on the lookout for these two species and I'll tell you how to identify them as we go through, but they're really large pigweeds. It's something we have to take very seriously in Montana. We're one of the last states without these two species. So why are we here talking about this? Well, according to the Weed Science Society of America, they are water hemp is the 5th most troublesome weed in the United States. Palmer amaranth is the #1 most troublesome weed in broadleaf crops, fruits, and vegetables. So let's talk a little bit about their biology of these two species. They're C4 plants, so they're warm season species. They emerge when soils are warm, a little bit later than kochia, if we're looking for a comparison in Montana. They have a really long emergence window that can begin in May and go all the way to July and August, especially under irrigation in the state of Montana. So sugar beets, alfalfa, durum, things like that. We have to think about that really clearly in those situations. Produce 100,000 to actually, some people say 1 million seeds. And they have both species have a lot of herbicide resistance, which I'll come back and talk about that in a little bit. So this is the plant family. We're talking about the Amaranthaceae. And this is where these species reside. It's a really big family. They took the amaranths and the chenopodes and actually redrew them to say that they're the same family now. So the amaranth family has about 60 genera. There's 300 some odd, 350, 360 species within in the family altogether. So it includes a lot of important crops, horticultural things. This is Amaranthus caudatus. It's I forget, weeping something. It's about love is the common name. And this is an amaranth species commonly grown horticulturally. Quinoa is the genus Chenopodium, right? We might hear about it, even people trying to grow it in the state of Montana. So it's another one. It's a Chenopodium species, but within the amaranth family. So how do we, lamb's quarters is another one that ends up landing in this family that can look superficially like a pigweed sometimes, but I'll tell you how to separate that in a little bit. Russian thistle and kochia are also in this family. So it's a big family with a lot of troublesome weeds in it. But it also has a lot of important rangeland native plants that we also like. Crassinacovia lanata, winter fat, common across a lot of our range in Montana, also belongs to the amaranth family. So now let's start with thinking of our identification, and we'll go kind of down the line and into the amaranth genus. So how do you separate the chenopodes, the common lamb's quarters, from the amaranth, the pigweeds? It comes down to spiny bracts. There's no spiny bracts in the chenopodes. There are bracts that surround the flowers in the amaranth genus. So if you have kind of a spiny plant, like on the right, that's our redroot pigweed, occurs all over the state, you would recognize that as the amaranth genus. So now that we're in the amaranth genus and we think about that, in Montana, we have 8 species. There's about 75 species in this genus globally. Some species are very difficult to distinguish from each other. And we'll talk a little bit about that when we get into our early detection, rapid response sort of reactions. So we can be confident it's not Palmer amaranth or water hemp if the plant is prostate and it's a pigweed. So this is Amaranthus splatoides, what we commonly call prostrate pigweed. We see it all over the state of Montana. It's usually not a problem in crops, but you see it really growing on the edges of fields, hedgerows, stuff like that, where there may be some compaction issues. It's not Palmer amaranth or water hemp if the plant is a tumble pigweed with flowers in the axials. And by axial, I mean like your armpit. If that's the axial of us, we could think of that on the plant. If there's flowers coming out of those axials, like in this tumble pigweed, it's not water hemp or Palmer amaranth. They both have axils on the terminal part of the plant, or the end, not between the leaf and the stem. So once we get into the amaranth genus, there's a group of 10 species in North America that are called dioecious. Dioecious means in Greek of 2 houses, and that means that there's a separate male and a separate female plant. If any of you guys have been trying to grow hemp in Montana or anything like that, you'll know that even though water hemp is not at all related to regular hemp, they are both dioecious plants. So there's a separate male plant and a separate female plant. And then diversity in these, because there's a separate male and female plant, they have to actually outcross their genetic material. And all that genetic diversity means they can evolve very rapidly to form herbicide resistance. In Montana, the only dioecious species are potentially Palmer amaranth and water hemp. So if you have a big pigweed that looks like it has no seeds on one plant, a male plant, and a bunch of seeds on another plant, and they look a little bit different, then those could potentially be Palmer amaranth or water hemp, so separate male and female plants. So it's sometimes hard to tell, and you really have to get down and look. So when we're separating them, the monoecious species with both male and female flowers on the same plant, that would be our redroot pigweed, really common in Montana. Also slim amaranth or Amaranthus hybridus, those would be species where you could really identify both male and female parts of the plant. And that would be the anthers and the pistil, or seeds and some old male stamens that you might see sticking up off the plant. If there's a male and female plant, it's an indication that it's water hemp or Palmer amaranth. So when we're identifying seeds and flowers, that's a little bit too late in the game for us, unfortunately. We'd like to be able to identify these early so that we can manage them and prevent any seed escapes. So what are some of the other characteristics we can identify these 2 species with? Both Palmer amaranth and water hemp have smooth stems with no hairs. And that would be in contrast to redroot pigweed. Redroot pigweed, if you look at the picture that's on the left, you see those little fuzzy hairs on the stem? That's a good indication of pigweed. If you look at those plants on the right side that we grew in the greenhouse, they're very glabrous. They don't have they don't have any hairs on the stem. So that's one good indication that we might have a pigweed. Most of our pigweeds in Montana, the redroot, the slim, not so much the Amaranthus Powell's, it has almost no hairs, but one indication, if you find hairs, it's probably not Palmer or waterhemp. The other identifying features is in Palmer amaranth especially, you have much longer leaf petioles. So I put an orange arrow on the right side. That's pointing to the petiole. The petiole is the leaf stem, as we call it, and it is longer than the leaf, sometimes as much as 2 times longer than the leaf. So you can fold it over, and if the leaf is shorter than the stem leaf, that might be an indication it could be Palmer amaranth. Redroot pigweed, on the other hand, has shorter squat petioles on the left side. You can see if you doubled that back and folded it, the leaf would be longer than the stem petiole. Also, the leaves, for when we're in this seedling stage, it's really harder to tell, but a little Palmer am and waterhemp have longer leaves. They're more pointier maybe in a way, but redroot pigweed, the way I remember it and describe it, is redroot pigweed's kind of shorter and squatter compared to the Palmer amaranth or waterhemp. Here's another view of those leaves. There's a picture of folding that leaf petiole back over to see that it's longer. And then you can see in waterhemp, the leaf is much narrower than maybe redroot pigweed, but it does have a shorter leaf petiole. Okay, so those are some leaf traits. The inflorescences differ. Waterhemp is long and narrow. That's on the left side. Redroot pigweed is short and squat, and that's kind of on the right side. You can see the difference between those two. This is a photo from the 2020 when we found the first population of waterhemp in Roosevelt County, Montana. And there was actually both co-occurring redroot pigweed with waterhemp in that field. Quincy Law, our colleague in North Dakota, has also found as many as 6 pigweeds in one field and containing waterhemp and Palmer amaranth. Last year, we searched a field for Palmer amaranth that had at least 4 different pigweed species. So you have to really kind of learn to identify these species within the genus. So here's the Palmer amaranth inflorescence. It's very long, very skinny, very cylindric. And you can see in this picture, the inflorescences aren't coming from the axials. They're really coming from branches or terminal branches on the ends of the plant. So redroot pigweed has male and female flowers, the same plant. Waterhemp and Palmer amaranth have separate. The female plants tend to be a bit more spinier, and the male plants tend to be a bit softer to the touch. And realize that not every plant is going to make seed. If it's a male plant, it wouldn't necessarily make seed. The key is identifying these much longer inflorescences. The last tricky pigweed I'm going to talk about before we move on and talk about why and our response, why we're concerned and what our response is Amaranthus hybridus, slender pigweed. It's a monoecious pigweed with male and female flowers on the same plant. The inflorescence is more elongated than redroot pigweed, and it's more common than I thought it was before I started looking at all the pigweeds that I find. I should also probably go back in and add Powell's amaranth into this one. It's another one that can be easily confused because it has a slightly longer inflorescence, but it's a monoecious pigweed as well. So male and female on both parts of the plant. Noel Orloff and I, the lead plant diagnostician at the Scudder Lab, we could tell you about looking under the microscope and identifying the male and female parts of the plant. It's a little bit tricky. If you're in doubt about your identification, call your agronomist, your extension agent, or Josh Wagner, and he will definitely help you or get you in touch with the right people. And that was what I was going to say here. You know, a lot of times we send things in or do genetic testing that is available. And so, you know, it's not even an easy thing for expert botanists to always easily identify, especially when they may come out of hay samples or other things like that. So we utilize genetic testing to confirm identification, and get in contact with us if you think you have one of these pigweeds. So why is Tim telling us in this webinar this is a huge deal, and we're talking about we're nitpicking the identification of pigweeds. Well, it's because the species is Palmer amaranth and water hemp are both spreading closer to Montana. Palmer amaranth really started out in the desert Southwest, pecans, cotton, things like that. It made a move into Texas and the Midwest and then into the Southeastern United States. You can see the color coding of the years that it's spread. And then it's moved into South Dakota, North Dakota, Iowa, Minnesota, and moving in that direction, and now coming back towards Montana. I think the first year of detection was 2018 or 19 in North Dakota. Last year, they found it in 5 more counties. It doesn't mean the populations are spreading or getting out of control, but they're finding more and more of it. And we really need to also be on the forefront of this in Montana to keep it in check. Part of this is the distribution of water hemp in the United States. And it's a distribution of water hemp that's color coded by resistance to herbicides. So this is really the big issue of why we don't want to allow the species to either species, Palmer amaranth or water hemp, to spread into the state of Montana. It's because in the Midwest, there's been the development or the evolution of a lot of herbicide resistance, and those color codes indicate the most. I think the next slide is talking about a couple of these. So we're talking about 6-way resistant Palmer amaranth. That's resistance to 6 modes of action. And in water hemp, we're also talking about 6-way resistance, which is really difficult to manage. I don't have time in this webinar to go through, but in Iowa, 97% of populations are resistant to glyphosate of water hemp. Over 2/3 of those populations are resistant to a 10x rate of glyphosate. Someone joked recently, Megan Anderson, the Iowa State Weed Extension Specialist, the best way to kill Palmer amaranth or water hemp in Iowa with glyphosate is to set a full jug on top of it. That's how useless these things have become, but also to chlorosulfuron resistance, 2,4-D resistance, glufosinate resistance. We could go on. And even the development of the new 2,4-D and dicamba soybean systems in the Midwest have already led to evolution of herbicide resistance to those. So that's why we're really in the prevention phase of these. Here's a little bit of the picture that putting out a few more of these. This is 6-way resistant Palmer amaranth in Western Kansas. Don't think it's just a corn soybean thing. It's definitely can move into cereals, sugar beets, pulse crops. So I think we need to be on the lookout. So resistance to chlorosulfuron, 2,4-D, atrazine, paraquat, lactofen, and mesotrione. And you see glufosinate there? Oh, glufosinate did it? Oh, never mind. Arkansas Palmer amaranth found resistance to rates of glyphosate. And this was 2021. So that's why I'm up here talking about this a lot in this context. Here's another one, 2020 dicamba not controlling Palmer amaranth. So it's tough weed. This is really one of the best models we could have for early detection, rapid response, because it'll be really difficult to deal with if it establishes in cropping systems in Montana. So preventing Palmer amaranth and water hemp from establishing is absolutely vital for our economics, especially, and for our cropping systems. So water hemp's been found 2 times in Montana. This was the second instance. These guys did a great job out near Fallon. They saw this funny pigweed in their field, and that's the sample on the right side of water hemp. So you get a little bit more idea of what it looks like. It was actually they found 2 plants in the field, and it was sticking up above the sugar beets. They got in contact with their extension agent, and she did great job, sent it into the diagnostic lab. We got it tested, identified. These guys did some really good searching of their field, only found a couple plants, pulled them out of there, and they and cleaned up that field. How did that water hemp land in this sugar beet field? It could have come by animal, by goose. It was migrating waterfowl. It's definitely one of the vectors. And that may be the case here, given that it was only just a couple of plants that were found in the field. The first instance of water hemp was found up in Roosevelt County in Durham and irrigated sugar beets. And that population was much more widespread. You can see those narrow inflorescences standing up and sticking up in the field. Palmer amaranth has entered the state as a seed contaminant, but it has not been found or confirmed in the state of Montana. In North Dakota, it's listed as a noxious weed. It's also a priority 1 noxious weed listed in Manitoba and maybe Alberta as well. It's a restricted weed seed in the state of Montana. So what are the vectors that then spread this into the state of Montana? So seed contamination has probably been one of the biggest ones. Millet seed, anyone growing sorghum Sudan, some of these millet crosses, German millet, proso millet, any of those things, if you're a forage producer, you should really be on the lookout for it. Most of that seed comes from the Midwest. It's a very small black seed. It's difficult to separate in from millet seed because millet is also a very small seed. Horticultural seed is another potential avenue. And then bird feeders and bird feed that comes into the state is another big one. I'll show you a graph, a couple slides down the road. Feed, if you're bringing in hay from different places, especially the Midwest, North Dakota, South Dakota, Colorado, Kansas, it could be a definite contaminant in those fields. People during the drought who maybe brought in corn stubble from Iowa or from Minnesota, some of these places, that's definitely one potential vector. When we talk to our colleagues in North Dakota, one of the biggest vectors that has spread Palmer amaranth in the state of North Dakota is people using sunflower screenings after sunflower processing as feed for cattle. So buying that and then transporting it to an area and then feeding it, that's been one of the other vectors. One of the other major vectors has been equipment, used equipment. So bringing in equipment, especially from the Midwest again, you really clean it as best you can. You'll never get every seed out of the equipment, but really be cognizant of that and clean the equipment as best you can. Custom harvesters who are traveling south to north especially on the small grain harvest of wheat and other things like that, bringing it in on equipment from that direction is another potential vector into the state of Montana. So look at your millet seed, especially in North Dakota, most cases of Palmer amaranth linked to contaminated millet. Contaminated millet seed has been identified in eastern Montana. Some of it was diverted, some of it was planted, and some fields are still being monitored in the state. I'll just say, as it goes out to be planted, I realized last year by going out and looking at some fields with other colleagues that when you plant something contaminated seed, and this was a 400-acre field, and you try to search a 400-acre field that has multiple pigweed species, it's a very difficult task. So ideally, you want to have really clean seed, make sure it's tested, and know about that before. Realize that it could be a possibility. Look at that seed label and realize it's a restricted weed seed in Montana. So let's come back to bird feed. I know I don't have a whole lot of time, but let's just come back to bird feed for a minute. There was a study in 2020 in invasive plant management science. 98 separate commercially available bird feed mixes were examined. Pigweeds were found in 94 of 98 bags of bird feed. Palmer amaranth was present in 27 of the 98 mixes. Pigweed species present in bird feed mixes included waterhemp, Palmer amaranth, redroot pigweed, smooth pigweed, and tumble pigweed. Not to mention ragweed, kochia, grain sorghum, wild buckwheat, common lamb's quarters, and a whole bunch of other things. So 5 of the mixes contained waterhemp and Palmer amaranth seed that were resistant to glyphosate. So we have one vector that's coming in very focused on our production agriculture, but we shouldn't forget about bird feed coming in the other direction. That might be something that may allow population to spread away from a residential area or an area where people are using bird feed. So what do you do if you get a suspicious pigweed? I say contact your local extension agent, Josh Wagner, or your agronomist to help you identify the plant. If Palmer amaranth or water hemp is suspected, a specialist will visit the site. We make confirmation, send it into the Scudder Diagnostic Lab at Montana State University. They identify plants. We'll want to know the GPS location, make sure we know the size of the population and how big it could be. Right now, we've been really sending it in for genetic confirmation. So we're doing some genetic testing to make sure that it's water hemp or Palmer amaranth. And then we really want to work with everyone and collaborate to determine the source. And most importantly, develop an action plan that allows us to contain the site. You know, we want to make sure we get those plants out of there. I'll come personally rogue plants for you. We'll develop a herbicide program if it's in a crop. We'll talk to the other weed managers in your area to get help and all those things like that. This is really a species we want. These two species, we want to stop both of them from spreading in the state. Here's a QR code link to the Palmer amaranth MontGuide that's available on msuextension.org. There's also a water hemp MontGuide that's in there as well. I'll leave that slide up for a minute and we can talk. And then I guess the last slide I have is just my question slide or my questions, comments, and if any of you guys want to do that now or we want to wait until a little bit later. Oh, I must have had some timing issues in there. So I'll leave it at that. I see there's a couple questions in the question and answer, and I'll try to answer those as I hand it over.
Okay.
So, yeah.
Tim, you got 4 minutes, so why don't we get a couple questions going?
Okay. Yeah, so if anyone has any questions, you can put them in the question and answer, or so I see there's a couple in there. Would I be able to get a copy of my slides to refer back to? Absolutely. We'll make this. Wonder, you could email me personally on my timothy.seipel@montana.edu email address, and I'm happy to share a link. And then another question, will there be a watch out for this weed flyer that's available? The MontGuides are available online. There is a mailer that will go out with to some of the seed dealers in the next few weeks that might be out there. And yeah, we'll put that out. We'll make sure that there's some mailers out there. But if you have any questions, send me an email and I'm happy to share any of the resources that I have with you. Oh, so Jane Mangold, my colleague on the other side of the wall over there, asked a really great question. If someone finds a plant, and she knocked on the wall, if someone finds a plant, should they pull it and take it to a local expert, or should they leave it in the field and leave it in place? This was a question that came up with some of our North Dakota colleagues. And I think in general, leave that plant in the field, maybe take some pictures or take one sample or two samples. A lot of times if the plant starts to wilt or you put it in a bag or it gets lost, falls into the bottom of your truck or something like that, then it gets harder and harder to identify. It's nice to know also the type of situation the plant is growing in. So in general, if you can, leave it in the field. And I promise that we will be a rapid response team that will come quickly and identify that plant. But in some ways, it's better left in the field and we'll come as the experts to the field. Any information on soil longevity? That's a really good question. The soil longevity studies say about 3 years. So 3 years of longevity in the soil, the seeds will be there. So that's what the state of Minnesota uses as their guide for monitoring. So Minnesota will say a population of Palmer amaranth has been locally eradicated if they monitor for 3 years after the event that might have produced seed or seed contaminated seed was introduced. They monitor for 3 years after that to make sure no Palmer comes back. What part of the plant? Oh, that's a good question. What part of the plant should be sampled? So I think it really depends on the stage. And actually, we can get genetic material out of pretty much all parts of the plant. Sometimes the stem is difficult to get DNA out of, but we can really use all of it to identify it genetically. If we're trying to identify it based on characteristics, we need to have a seed head, the full inflorescence, and the full part of the plant to really evaluate it. Because we evaluate not only the leaves, the stem hairs, things like that, but then we really start looking closely at the inflorescence, the length of the bracts, a lot of very complicated botanical features. So I would say we'd want all the plants. Without it flowering, it's really hard to identify it just based on vegetative characteristics.
Thank you, Tim. That was great. I thought half an hour would be plenty of time, but that went by really fast, and I'm sure there's so much more you could share with us. But we will go to Alice Whitelaw and Ngaio Richards from Working Dogs for Conservation, and they'll have half an hour, and then we'll have 10 minutes of questions for both Tim and Working Dogs. And then if people want to continue to talk or whatever, I can stay on as long as possible and answer questions as well, or chat about anything. So please go ahead, Alice and Ngaio.
Sorry, give me one sec. There, can everyone hear me okay?
Yes.
Fantastic. Okay, well, thank you very much to Tim. That was really, really interesting, a little scary too. And I want to thank Josh for pulling all of this together. We've been getting increasing interest and questions about getting dogs on various plants. So we thought it would be most efficient to try and get as many folks and interested parties on this call, as possible. And so right off the bat, I wanted to mention that in preparation for this, Alice and I got on a call with Josh, and we went through each of the plants on the Montana Department of Agriculture Early Detection Rapid Response Program page, and that was a few weeks ago. And so we considered the state priority criteria, and we matched that up with the criteria that we consider around the feasibility of getting dogs onto plant targets or any other target for that matter. And so those criteria and the thought processes are the underlying aspects that I'm going to be talking about here before turning it over to Alice. But I also wanted to say, not only was it fun talking to Josh, it's always fun talking to Josh, but to the question of how and why might we get dogs on a plant target, with us, it really starts with an exploratory conversation, and that's typically with Alice. And before I move on, I just also want to note that this handsome blonde fellow is Tobias, Mr. Tobias, and he's posing after a very successful search session in Sweetgrass County. And sorry, and this picture was taken on what I would characterize as one of the best days of my life. It's a beautiful location. The weather was beautiful. We were out with some great people. It was an excellent day of work with a very fine dog. And what could be better? And I'm not just being sentimental. I also mention this because one of the perks of working with dogs, can be very overwhelming working with and trying to get a hold of some of these invasive species. So the dogs are always there for us in that way. So here's what we're going to be talking about. Plants and dogs. And I'm going to end on Palmer amaranth. We're going to come back to it. But first, I want to revisit, Josh listed these in his intro, but just to reiterate and also to provide a visual, because we humans are such visual creatures. So the priority 1 plants are those that are not present or have a limited presence in Montana. So we've got yellow star thistle, so left to right, Dyer's woad, purple loosestrife, rush skeleton weed, Scotch broom, and blueweed. And then with your priority twos, those are common, but in isolated areas of Montana, or they aren't abundant in many counties. And so again, left to right, we've got tansy ragwort, yellow flag iris, flowering rush, and salt cedar. Some lovely EDDR grasses, medusa head, common reed, and ventinata. And then we just talked about water hemp, common bug loss we haven't talked about yet, but last but not least, Palmer amaranth. And I won't tell you how many times I had to say it to be sure I was pronouncing it correctly. It was very helpful to have Tim go first and rattle it off a bunch of times. So, sorry, just to go back for a sec, we're going to touch on all these plants in one way or another. And like I said, we're going to conclude with our now very good friend, Palmer amaranth. But I'm also going to raise some examples of plants that aren't mentioned just to kind of show, convey a little bit more some of these applications and how the dogs come into play. I'm going to lay the groundwork, and then Alice can answer questions and provide more detailed information about things like training, dog selection, associated costs, and other important logistics, whatever people want to know more about. And I'm sure folks will also have questions for Tim and Josh too. So getting back to Dyer's Woad, it's kind of become the poster child for dogs and plant work in Montana. And most people are familiar with this success story. We're really proud of it. But because it's so well known, I'm only going to be referring to it in passing as we go through various plant examples in order to highlight just what it is about Dyer's Woad itself and the approach that led to the successes relative to this of the dog team efforts. But I do want to take a moment to thank, I don't know if she's on the call, but any chance to sing her praises. And sometimes it makes her blush, which I take pleasure in. So, just huge thanks to Amber Birch for her vision, and to Marilyn Marler and Tris Hoffman, just for their unwavering support and efforts to ensure the longevity and the traction of this work. And again, not to be too sentimental, but it's a pleasure to be part of this collaborative team effort. And it really is. Everybody is just fully engaged and working very hard. So it's why dogs? I mean, why? It's no secret that dogs, they have phenomenally good noses, and they're better than ours by a long mile. But I think people often don't realize just how superb the dog nose is. So we often will seek to quantify that a little bit. So an example we could give to help visualize that concept is that if you were to remove all the scent receptors in a human nose and lay those end to end, they would fit on a surface the size of a postage stamp. That's compared to the size of the area, the number of scent receptors in a dog's nose would occupy end to end. Those would fit in a space the size of a football field. And in terms of detection thresholds, we can think down to a cup of sugar in an Olympic-sized swimming pool. Now, for our purposes, the dogs learn from us during training to associate the target that we want them to find. So in this case, it would be a plant, and communicating to us when they find it. And they do that by performing an alert, which is typically a sit or a down. It's not always. And in return, they get a coveted reward, and that's playtime. And there are some very important considerations to add, like how on the one hand, not just any dog can do this work, that's conservation detection work, that's also plant work. Actually, very few have all the requisite characteristics and traits, one of them being an intense toy drive. On the other hand, we look for candidates on the basis of them showing certain traits rather than favoring or seeking out specific breeds. So we often end up working with certain breeds, but we're really looking at those individual traits. And many of our working dogs are in fact former shelter dogs. So these are all things that I just wanted to mention, but we can explain more during the Q&A session with Alice if anyone is interested. Oops. The list of plants you see listed here is a selection of ones that dogs, not just from our organization, but others as well, have either previously been enlisted to help find during active fielding, or been tested on to see if they could distinguish the scent, or discriminate one focal plant of interest from several similar plant species. Now, I hasten to add that this list isn't exhaustive by any means, and just these examples were selected so that we can discuss some of the factors that make a plant target conducive or challenging for detection. Sometimes the same plant is conducive but also challenging depending on the life cycle stage that you're looking for. Now, if she isn't fast asleep, but she may well be, and Josh mentioned her too, we have our dear friend and colleague might be on this call, and she's based in Tasmania, and she's worked on orange hawkweed, and she's already worked on several grasses, and we here are getting ready to work on grasses for North American applications. Now, dogs can be trained to multiple targets and to seek them simultaneously, which is sometimes news to some people. And they also, again, they have the ability to discriminate similar types of plants or species one from the other. So that can be really useful in this kind of work. So for example, and what you see on this slide is a graphical representation of one of the handler's field notes, and that includes the exclamation marks and the understated enthusiasm. In the Neil Smith Refuge in Iowa, dog teams started out by surveying for invasive Lespedeza, and then a few years ago, the same dogs had whorled milkweed added to their scent repertoire as part of efforts to assess the habitat quality for monarch butterfly. And a major finding from this work was that the multiple prior years of surveys were really helping to knock back the invasive Lespedeza. And at the same time, whorled milkweed was being found all over the reserve, which managers hadn't anticipated. And the last piece of the puzzle is that the dogs were able to distinguish the invasive Lespedeza from the native Lespedeza, so only telling the handlers about the invasive plant. And this multi-target application is a great example of how we can sometimes be a little bit categorical and linear in our thinking and even on our management strategies. But it's worth bearing in mind that fundamentally, dogs, they don't care whether a plant is native or invasive. They just want to work and find their target and be rewarded for that find. So as your monitoring and eradication takes hold, there can be some benefits to providing a dog with additional opportunities for reward, like in this case, while the Lespedeza is being dialed back, by layering on a complementary target. And you aren't just gathering information about eradication at that point, but you're also gathering data potentially about habitat quality. So we sometimes think of weed management and conservation efforts separately, but they're very interrelated. And the fact of dogs being able to search simultaneously, sometimes for an imperiled species or a native species or an invasive plant or weed, can create opportunity for partnership and resource pooling between the two. So say, for example, between weed managers and conservationists or researchers and preservation entities. And I'll say this a few times, but the handlers can also offer some unique expertise and capacity. So the stray violet example I had in the last slide, and I'm sorry, I can't quite recall what species it was. The dogs were trained to that violet species, also in the Neil Smith, if memory serves, but they didn't seem able to find it once in the field. That wasn't successful. But the handlers could keep an eye out for it, and they did manage to observe a few and record those locations, all the while supporting the dogs in finding the primary targets. So the handlers, we do a lot of things, and that can include looking out and gathering a little bit of extra information supplemental to what the dogs are doing. Another example of discrimination arises from the Kincaid's lupin work. And this is where the dogs were able to discriminate Kincaid's lupin from other lupin species that were present. And this is an application where data can be indirectly gathered about the imperiled Fender's blue butterfly, which favors Kincaid's lupin. You can get a sense, I think, from the photo of how challenging these plants can be to find without the dogs. And when I finish my part of this presentation, Josh is going to show a video we sent of a dog called Lily working. This pretty girl here is with Camus. But so you'll be able to see how it looks to have the help of a dog to find that proverbial needle in the haystack. And at this point, I also want to just interject that dogs have the capability to be trained to recognize and find multiple scent targets, not just a single target or one type of target. And Alice can explain better, but the number of target scents in a dog's repertoire is strategic and based on the complementarity of targets to one another and often the dog's main deployment. So in conservation, as in other detection fields, there are many good reasons, an abundance of good reasons why it might be necessary that a dog only be on a single target, much like the case with explosives dogs, for example. But I just want to impart that the dogs are certainly capable of learning and finding more than one target. So with time, we have learned that not all plants, or at least not all life cycle stages of plants, lend themselves equally well to dog detection. We did a short opportunistic pilot study on yellow star thistle, and what we found overall was that at the spiky flowering stage, and as the plant got progressively woodier, the dogs had to be almost upon the plant before they smelled it. And at that point, we could see it, and sometimes we would see it before the dogs appeared to scent it. And it did also seem like the dogs were able to discriminate star thistle from other plants at the small leafy rosette stage, but we kind of ran out of time to fully pursue that. But it does something that Tim mentioned a lot about the importance of early detection and rapid response. The finding or the observation about dogs being able to recognize the early leafy rosette stage does point towards some potential applications for early detection and at the tail end of that for assessment of the success of eradication efforts. So that could be really useful, even if at the flowering and spiky stage, it's not necessarily a very cost-effective use of dogs. So just thinking back to the Montana list that Josh mentioned and that I went back to for similar reasons, other plants that come to mind as possible candidates for early detection might be rush skeleton weed, Scotch broom, and blueweed, and of course the pigweeds, which I'll get to in a few slides from now. And so at the exploratory stage, this is where assessing human efforts relative to dog team performance really comes into its own. Sometimes we're in the field and we joke a little bit about having a healthy competition between the human teams that are out looking and the dog teams. It's so useful to be able to determine when or if, along that spectrum from early detection to rapid response to eradication, if the use of dogs might be optimal and crucial. And the way or the stage where the dogs might turn out to be the most effective isn't always the one people are expecting, especially when you're exploring a prospective plant target. And again, Alice can speak more to this, but I just wanted to point out here that the way that you would meaningfully measure performance and what the dog teams might be able to offer relative to the ongoing human efforts is that you would compare the way that humans operate best to the way that the dog teams operate best, not trying to get dog teams to execute a search the way that people do, because the search is typically executed, well, it's going to be different because we are very site-based and the dogs and the dog teams are operating on the basis of scent. This is the handsome Tobias again. So how do we train? Well, I'll let Alice speak to this more if there are questions. But like I said, our dogs associate finding the desired target with getting a reward. And these dogs are selected for being intensely toy and play driven. So, but they also, another thing that I hasten to mention is that they also want to work with a handler, which is what for us is one of the very meaningful aspects of the work itself. But not all dogs who, even if they show tremendous olfactory prowess, or they're not necessarily going to want to work with a person. So the first step is to give the dogs exposure to the scent itself, for which we need good representative samples. And once we reach a stage where the dogs are showing scent recognition, we then need access to training sites so that the dogs and really ourselves, we can all problem solve in the field, because there is typically a steep learning curve, or at least a learning curve, once you move away from the controlled training scenario, where you're basically presenting in a fairly straightforward manner the scent to the dog. And then once that you get to where the target occurs naturally, there can be all kinds of impediment to the way the scent gets out to where the dog might be able to encounter it. And a critical parameter that really comes into play in situ is detection distance, as in from how far away does the dog show what we call a measurable change of behavior, and how far away do they therefore need to be from the target in order to detect it and work towards the source of the odor, because that is the core piece of information we need to then plan our search strategy. Things like distances between transect lines for good coverage. And before, when I was talking about comparing human and dog team performance, one of the parameters we consider is detection distance, not just relative numbers of finds, like who finds more when they go into this area. Similar to the slide where I was talking about the simultaneous search for Lespedeza and whorled milkweed, I wanted to share this image to give you a greater sense of how we might divide up an area we've been asked to monitor for a particular plant or plants. And that's working very much with the established detection distance and other environmental factors like wind, heat, vegetation, other vegetation. And this is what it looks like. It's a dog and a handler, sometimes with an orienteer, sometimes with a few other people in tow. We're working, we're out with our dogs, and they're leashed or unleashed according to requirements. And I honestly don't mean to romanticize it, because it's often hard, grueling work, but there are a few more enjoyable things than having the light come up on you while you're in the field working with a dog. And a good team, and this is a good team that's pictured here, will make it look deceptively easy, like they're just out for a stroll, when in reality there's all kinds of factors coming into play. So some important considerations around using dogs for plant work are density and visibility. And we did some work on knapweed, and the dogs were very proficient at detecting it, but it is absolutely everywhere, unfortunately. Now, that doesn't necessarily need to be a limiting factor to enlisting dog teams, but it's usually going to be far more cost effective to have people reduce the population and then bring dogs when density is reduced. And practically, it can be very overwhelming for the dog when they get into an area that's just chock full of their plant target. Density is obviously different from visibility, but even with something like blueweed, which isn't terribly difficult for people to differentiate, because it's got those characteristic spots on the stems, the way we divide up a search area to work our way through with the dogs, and that's, like I said, based on factors like detection distance, environmental factors, it just inherently makes it strategic. So we can get that comprehensive coverage. So you can get plants hiding in plain sight that you might overlook sometimes. And for something like yellow flag iris, the plants can be very conspicuous when they're flowering, but you can still have varying stages of growth within a patch. So if you were to pull the flowering plants, one way to use the dogs would then be to return after a certain amount of time to see what's still there. And that's a strategy we've used with Dyer's Woad, often within the same season, to tremendous effect. And purple loosestrife offers a similar example. It's a riparian plant, and it's got a distinctive square stem, which is notable. But even if it's obvious as a flowering plant and pulled, there could still be seedlings that get overlooked. So we also need to be mindful of not removing native vegetation if possible. So like I said, and this is very much from experience, some plants can absolutely be hiding in plain sight, and you wouldn't know to look at it. But this flowering monster was tucked away in a recessed area Mount Sentinel. So for any of you who've ever been up there, there's kind of, the best way I can describe it, there's like little pockets where the hill kind of folds in on itself. And this is where this big whopper was lurking. And I, thanks to the dog. Now, on a peripheral topic, I can also say that one of the neatest contributions, which I can't overstate, and which arises due to the scent versus visual focus of dogs, is how they bring us into unexpected areas. So surveyor bias is a real thing. And searching only and increasingly where you've previously made finds is what it amounts to. But searching by scent, I could give you several different examples where dogs showed us that the target doesn't occur necessarily where we expect it to. So that keeps us humble, but it also, in practical terms, it also ensures we aren't inadvertently restricting our search area and missing something we need to stay on top of. So Tim was mentioned genetic testing, and this is another amazing thing we learned from the dogs. And I do think this is somewhat unique to Dyer's Woad, that how differently the rosettes can present visually at different sites. If anybody's interested, I started collecting pictures for sort of a Woads of Montana gallery that I'd be happy to share. And it really shows the range of rosette appearances across our sites. It's kind of incredible. But it's down to several dogs that we realized this phenomena. Amber and I were out, and the dog we were with was absolutely insistent and sitting at this plant and making all these indignant noises, but the plant didn't look woady at all. But we grabbed a sample and she had it genetically tested, and lo and behold, it was Woad. So seems like the variation in rosette shape is due to different drainage patterns. But because we know that from the dogs, it helped me and it helped us adapt our search pattern so that we now look for different visual iterations of the plants. And we also know to recognize when the dog is telling us, we know it's a Woad, even if before we might not have thought it was. So I'm winding down, and I wanted to share some related canine scenting applications to ponder, just to give a sense of what might be possible. And especially after hearing Tim speak, I'm looking, I don't know if you can see my mouse moving, but I'm looking on the bottom second from the left at the equipment inspections, seed inspections, bird seed inspections, potentially as just different ways that dogs might be able to help in a discrimination capacity. I think we all understand that people like dogs, love dogs, and yet they do have a very legitimate and often overlooked ambassadorial role to play. Now, I happen to adore plant work. I'm passionate about invasive species, and I'm passionate about that with and without dogs. But I guess not everyone thinks that is as sexy of an application as I do. However, the mere involvement of dogs in invasive species monitoring has garnered millions of dollars worth of free media publicity. That's in Canadian dollars, but that's also in American dollars. So thinking about zebra and quagga mussel interventions, and also emerald ash borer, just as examples to provide. And dogs also have a way of facilitating engagement and compliance like no other. And when this is harnessed for conservation and management efforts, it's a beautiful and quite powerful thing. And speaking of beautiful, dogs can certainly be incorporated into outreach and educational materials. Here's some children's materials that we were working on or have in the works for Dyer's Woad. And, thinking about something like tansy ragwort, which can be spread, I learned from Josh, from logging and firefighting equipment. So dogs could be incorporated into or assessed to see if there's feasibility to incorporate them into equipment inspection, into various outreach efforts, either having a meet and greet where you've got a dog on hand and then you're just talking about ways to minimize the spread. And I would argue, again, just trying to minimize that linear thinking we sometimes engage in. I would argue that the dogs don't necessarily need to be trained on the specific plant to be brought to a meet and greet, but they could be trained to other plants or applications. But it's just a great way to generate engagement about the current application. And purple loosestrife is another one that comes to mind for outreach, say with beekeepers who might be invested in the importance of a non-native plant like purple loosestrife as their livelihood centers around it. And so generally we found sometimes people who might otherwise be quite resistant will soften a little in the presence of dogs. And it's fun to get a little bit creative with something like purple loosestrife, thinking about developing specific outreach tools, including pollinator information about pollinators and graphics, and factoring in dogs, just to reach more people and different demographics and different age groups. So again, there are various possible applications, and I say possible because we've learned an enormous amount from our work on Dyer's woad and some of the other plants we and others have worked on, but there is still quite a lot to be learned in the realm of plant work, whether that's invasive or native. And I would argue that, given what we do know about the difference dog teams can make when a target is viable, it's worth going through the motions on what seem like promising targets, even if only to rule them out and to gain more information about just what it is that is not making them lend themselves well to dog detection. And again, measures of dog team versus human find and detection rates are incredibly useful to gather in this regard. And it's not just a matter of whether dog teams find more as a tally. We want to be thinking about whether dogs are instrumental in early detection and rapid response, just due to their ability to discriminate at what I would call the inscrutable rosette stage. Are dog teams able to help find more plants before they reach the dreaded flowering or seeding stage? That was the case with Dyer's woad. Accuracy to target is another parameter we look at, especially when you're doing a visual assessment. For example, does the involvement of the dogs ensure less of a native subspecies is being removed by the dog discriminating to the invasive plant and ignoring the native plant? And just quickly to end up, for those of you who have dogs, I always like to share the existence of this trusty field guard, which really does help protect them from seeds in their eyes, their ears, and their noses, their mouths. It's also important to mention here that we ourselves never want to become vectors of invasives as we're doing this work, and we take great pains to avoid that by doing rigorous body checks once we get back to the vehicle, and then also once we get back to whatever accommodation we happen to be staying in, which is not just for the fundamentals of the work, but also for the dog's safety. And there will be some plants that are detectable. A lot of times the question isn't, can the dog detect it? Can the dog recognize the scent? The question is it operationally feasible? Is it realistic? It could be simply too hazardous for a dog to do the work if they occur in an area with hazardous vegetation or in hazardous and impenetrable terrain. And there could also be ways around that to mitigate hazards, or again, looking at something like medusa head or ventonata, can the dogs detect those at a very early stage when they're a little less pokey, to use the technical term. And another thing I'd like to mention is that the ideal timeline or time frame for dog team surveys might differ from the usual or what people have in mind. For example, if we need to be out when the high water mark is lowest, although dog teams can work from boats and travel between survey sites by water, but we also might wish to go out when the scent is most abundant, and that could be well before the more evident flowering stages around which some human surveys or volunteer citizen science days might be scheduled. And again, going back to what I was saying about comparing performance, this would be an example where you might compare outputs from, I don't know, autumn flowering plant surveys and polls with people versus outputs from spring surveys with dog teams. And you might find that incorporating both really boosts monitoring and eradication efforts. It's almost never just one or the other. So like I started this presentation by saying, if you've been pondering dogs or how they might be incorporated relative to a particular plant or plants, the first thing would be to chat with Alice and in coordination with Josh. And typically we and our counterparts do operate on a fee-for-service basis, but as we heard, now we get back to our good friend Palmer amaranth, to which there is simply zero tolerance for this plant, especially after hearing what Tim had to say. That's the lesson learned from other states and wanting to avoid the plant's occurrence at all costs through prevention, through vigilance, through early detection. So given the stakes and the potential scale of that, it might instead be worth considering the development of a program to have unrestricted access to a dog team or dog teams year round. And for just, again, to give you a sense of how it might look, it could entail something like deploying dogs across the state to see if they can pick up on the petiole stage plants before it goes to seed or is more visually obvious to people. So Josh is going to show you the video of Lily working, just so you can see a lot of what I've been talking about kind of come together. And we have a large repository of reports and papers, some of which we've co-authored, others authored by folks doing great work in other organizations. And if you ever wanted to have more technical details about plant detection or more information about the dogs and the people who do this work or the olfactory science, other conservation dog applications, please reach out. We're always happy to share. And with that, I'm going to hand it back over to Josh, and I'm going to thank you again very much for the opportunity to talk about one of my very, very favorite subjects. And I have no idea how much time I've taken, so hope I didn't gobble up too much time.
Thank you, Ngaio. I'm going to quickly share a video of this is Lily. Looks like it's Tobias, but it's not. It's Lily going after world milkweed. Is anybody else smiling like I am now? So I don't think that picture or that video quite shows one of the really fun parts is when the dogs do get so excited when they get their toys. They just go nuts and they want that toy so bad. It's amazing the work they do for it. So before we ask questions, I just kind of want to wrap up from my end. Thank you to Working Dogs. It's one of the most enjoyable parts of my job working with them. And in addition, they are super effective, like discerning really, really tiny rosettes. Whether or not it truly is the plant, humans can't always tell, but the dogs can. but they are great ambassadors. And when I'm out presenting across the state on these different weeds, people are always so interested in the dogs, and it just opens the conversation. And even though I teased about being international, because Niall's from Canada, they're also a very Montana organization. And then thank you to Tim and MSU. Everything I do. Greta's messing with my computer, so I'm kind of trying to see what she's doing. Anyway, every day the research and education stuff that MSU gives us, I'm using, and just a wonderful free service across the state constantly. And if it's not Tim with these weeds like Palmer amaranth, you could have Jane Mangold and Lavrite Shergill and Noelle Orloff, and all helping to fight this battle. So just a big thanks to MSU. And I will let you guys answer some questions. So on average, how long does it take to train a dog to reliably identify a given species, and how much variation is there across dogs and the species they are being trained to detect?
That's a great question. This is Alice Whitelaw, and I'm the director of programs here at Working Dogs. The dogs, particularly a dog that has already understands the game, that's already been deployed on, even if it was something like a carnivore, it only takes a matter of days for them to learn a target. But to be able to reliably identify a given species takes a little more time, because then you're moving from that very controlled scenario where you're introducing the scent and giving a reward for correctly alerting to that scent. When you get out into other vegetation and the complexities of given landscapes and environmental conditions, it takes a little more problem solving. So we've successfully trained dogs to find a brand new species of plant within 2 weeks. And that has also, where we are able to field them then. Within that 2-week period, we introduce the scent, and then we are able to determine detection distances, which help us figure out what the transect width is going to look like, or the search strategy is going to be. So it can go that fast. And then there is variation between dogs. Some dogs aren't very good at detail, and plant work typically is detail. So those are dogs that we wouldn't put into that situation. So it's an individual thing.
I had a question for Tim.
Yep, I'm here.
Hey, I was just curious. You were talking about detection of Palmer amaranth or water hemp in seed mixes and bird seed, and I just wondered how that was being tested at the moment.
I don't think there are any laws governing bird seed, and I'm not sure that any of it is really tested. We don't actually test it. That was just a study that was taken from, they just went around to the supermarket and bought a whole bunch of different bags of bird feed and looked through it themselves. When it comes to seed inspection, all of that is really done and packaged when the seed lots come in. Josh can answer some of those questions better. Ryan Solberg is part of the grain testing lab. They do a lot of that stuff. There's a seed testing lab at MSU where seeds come in, and that is basically, they sort through the seed samples manually that are taken and identify all the seeds by species based on their morphology. So it's a very labor intensive thing to try to inspect and look at and stuff like that. You know, there's a lot of seed cleaning machines out there, but you know, nothing is 100%. So there's a lot of contamination that was out there. I don't know, maybe Josh, or if Ryan Solberg's on today, they could jump in and ask or answer a little bit more about how the grain and seeds looked at a regular basis or inspected on a regular basis.
How about I say, if anybody wants to information on some of this stuff, details, shoot me an email afterwards, and I'll get them the information. If anyone wants to go down that orange hawkweed in Australia vortex like I did, I can share a bunch of that information. I can share some of the seed information. Really, whatever you want. There's a lot more here than we could cover in an hour. So just email me with that and I'll get details. But I also wanted to ask Tim a question. I just couldn't find my unmute. And that is, Tim, I mentioned that you're an excellent botanist, one of the best I know. How do you think you would do against a dog?
I have actually gone up against the dog before looking for knapweed out in Belgrade when I was a master's student. And I think I found 86% and the dog found 92%. It found a couple of rosettes that I could not see when then I was quickly searching over the area. So I like the combination.
I think we know each other then, because I was part of that project, and you might have gone up against Savo, a big gray German Shepherd.
Yep, it was, and that occurred in Belgrade, Montana,
Yeah.
about 15 years ago or so, I think.
2005, 2006.
Yep, there we go, even longer than that. Yep, I was a graduate student at the time, and I was part of the study.
That's awesome.
Mm-hmm. Yeah, I see the advantage of having the dog, but it's a really hard thing to find, you know, and yeah, searching is difficult in that. Yeah, walking into some of these millet fields with 4 or 5 pigweed species, I'm like, whew, wish I had a dog that would search this field. Nope.
With that, yeah, we can also, if anyone wants to send me an email, we can get you on a list to help us go rogue fields for Palmer amaranth if it shows up.
Yeah.
Any more questions? I think there's still 75 people on. We peaked at 90, which I think is pretty good.
I just had a quick comment, if I may.
Sure.
Yeah, I well, it's because I'm a big dork when it comes to dogs and plants and weeds. But one of the neatest things I have found with this whole aspect is the interplay of human efforts and dog team efforts. And like I was saying, and you know, Tim kind of alluded to, there's this fun competition. You know, we occasionally have we have a guy up at one of the sites who monitors one of the sites over the season, and then we come in the autumn and compare notes on what the dog found versus what he's been keeping tabs on. But it really is very much a partnership, and there's a role. There's plenty of work for everybody, and it's just interesting every time we look at a new plant species or a new application to see ooh, I wonder where the dogs might fit in relative to the already good work that's being put in. So I always like that.
I see that Ryan Solberg got added in. Does Ryan, do you want to say anything about the grain inspection processes or anything like that?
Yeah, this is Ryan here. Yeah, for the seed program, really what we're looking at, we have inspectors throughout the state that can do inspections and collect samples of any seed that comes into Montana, and we send that to the lab. And currently they're aware of this problem and looking at specific seed kinds that could be a problem and collecting samples of those. And then any amaranth species we're sending on to get DNA genetic testing to determine if there is Palmer. And so those are some of the steps that the seed program is taking. But currently, not all seed coming in the state is tested. It's routine inspections that we come across. So a big part, of course, is educating seed dealers of this problem. So it's good to be part of the task force and in doing that.
Thanks, Ryan. Ryan's got a big job with that. We appreciate his help.
I see there's another dog question. I'll go ahead and answer that. Is it just younger dogs that get trained? When we're looking for candidate dogs, we're looking for dogs between the age of 1 and 3 years of age. But once a dog has been trained to a target, no matter whether, like I said before, whether it's carnivore scat or plants or invasive mussels, even that dog can then even past the age of 3 all the way up until the time they're 12 years old, learn new targets. And we've had, I think our record holder was a black lab that had 31 targets on her. I think the record I have with my own personal detection dogs was, I think, 14 targets. And that varied from plants to live animal to also carnivore, multiple carnivore species, scats, just to name a few.
Okay, going once. We know all of us are busy in the weed world and don't need to sit here all day, but these are, like I said, some wonderful experts that are nice to take advantage of. But if there's no more questions, I guess we'll wrap up, and thank you for attending, and please send me an email if I can share any of this information better that happened today, and look out for our fall webinar. Thanks.
Thanks, everybody.